So cell, the unit of life. So in this chapter, we'll be learning about the two types of cell basically, that is the prokaryote and eukaryote, as well as what are the organelles which are present in them and its functions. Fine. So starting with the first thing, that is what is cell. So this is a basic thing. Okay. If that is cell. Cell means what? It is nothing but the fundamental, structural, and functional unit of living organisms. Okay, so this term, that is cell, it was first time coined by Robert Hooke. Okay, whereas he termed this thing, that is cell, but whereas the first ever living cell, it was seen under the microscope by Anton van Leeuwenhoek. Okay, so every cell has a major structure in it, which is called as nucleus. So the term nucleus, or this nucleus, was discovered by Robert Brown. Okay, so these are some important persons. Okay, so next one is now how the cell came, what are the types of cell, on what basis the two types of cells are there, that is prokaryote and eukaryote. So this is all based upon something which is called as cell theory. Okay, so this cell theory was proposed by two persons, that is Schleiden and Schwann, in the year 1838. Okay, so what is cell theory? Very important. So cell theory means nothing but, first of all, it says that all the cells, okay, they are made up of what? More than one or more cells. Like for example, we say unicellular, multicellular. Okay, so that is the first point in this theory, that is all living organisms are made up of one or more cells. Okay, second one is what? According to the definition, that is cell is the basic structural and functional unit of life. So that is the second, the theory. Structural and functional unit of life. And the third point of this theory is all the cells arise from pre-existing cells. Pre-existing cells. So these are the 3 points which was given by Schleiden and Schwann under the cell theory. Next is size and shape of cell. Size and shape of cell. So now there are even two types of cells, if you can say, according to the size. One is the smallest one, another one is the largest one. And coming towards the shape, there are different shapes of cell, okay, which we are going to see. So first of all, regarding the size of the cell. Okay, now all the cells, they are like in variations. Okay, you cannot say that this cell is same as the another cell, because the size will be different, the shape may be different. That's the reason. Okay, not only size and shape, but also whatever activities they do, or the lifespan means how many days that cells will live. Okay, so all those things, according to all those things only, there are different types of or shapes of cells. So the first one is the mycoplasma or PPLO. Okay, so PPLO means pleuropneumonia like organism. So this is the smallest cell. Smallest cell. It's about 0.3 micrometer in length, approx to 3 to 5 micrometer in size. So this is the smallest cell, mycoplasma or PPL. Second one is the largest one. So largest egg, a single cell egg, which is ostrich egg. Ostrich. Okay, so here about the size, if you measure it, it's about 170 to 135 millimeter. Plus you have what? The RBCs, okay, the human RBCs and the nerve cell. Okay, so in case of human beings, the longest cell is the nerve cell. Nerve cell is what? The longest cell in human. Okay, so this is all about the smallest size cell and the largest one. Next one is the shape of cell. So shape, there are many shapes of cell. Okay, like it depends upon the bacteria again. So you can say they are cocci. Cocci means round shape. Then bacilli, which is rod shaped. Okay, then you can take spiral. Okay, that is like this, spiral. Then vibrio shape, which is somewhat like this, comma shaped. Also, it's called as comma shape. So these are what different shapes in which you can find the bacteria or the small microorganisms, you can say. Now over here, the cocci, bacilli, whatever is there. Now it depends. Like it may be only one, okay, cocci, or it may be diplococci. Diplococci means what? Two cells will come. So this is called as what? Diplococci. Or else it may be a staphylococci. Staphylococci means many will come like this, rounds. Or a streptococci, which is spherical shape in a straight line. Okay, in the same way, you have it for the bacilli as well. That is diplobacilli, streptobacilli. Okay, like that. So depending upon how many rod shaped or how many cocci shaped are present, according to that, the names differ. But these are what the basic 4 types of shape which you can find in the bacteria. Okay, so the shape of the cell, it may vary according to what type of functions they perform. Fine. So this is about the size and shape of cells. Next one is the types of cell. So this type of cells is categorized on the basis of organization, the complexity of cell, as well as the variety of cell. Okay, so on basis of all these three, the cells are basically divided into 2 types, which is prokaryote and eukaryote. Prokaryotic. So pro means primitive nucleus or no nucleus, and U means true nucleus. Okay. And carrion means nucleus. Fine. So prokaryotic and eukaryotic. These are the two types of cells. So first we will see the prokaryotic cell. So if you see the prokaryotic cell, it's the most simplest form of cell. Okay, because it has only few components inside it. So I'll just draw it. Okay, just a simple cell I'm drawing. Okay, so this is just a basic uh, prokaryotic cell. So the most important parts over here, let's see the first one. Okay, so here we'll be seeing three things which is for the motility basically. So one, which is this blue color, which is flagella. Okay, this is what the flagella. Then this big thing, which is called as the pili, and the small hair-like structure, that is the fimbriae. Along with it, if you see this structure, this is called as mesosome. Okay, then these are ribosomes. Plus, it has some food vacuoles. Okay, then now if you see over here the structure, the black colored one and the green. Okay, so the first one which you have over here, this entire thing is called as what? Glycocalyx. Glycocalyx. Then you have here in between, which is the plasma membrane. And then here, this is the cell wall. Plus liquid-like substance which will be present inside the cell called as cytoplasm. So these are what the few things which are present only in prokaryote. So apart from this, extra things will be present in the eukaryotic cell. So based upon this only, we are going to see the, the characteristics of the prokaryote cell. So the first thing about the prokaryote cell. Now, these examples which you can consider over here is bacteria, BGA, that is the blue-green algae, mycoplasma, and so on. Okay, so basically these the prokaryotic cells which are there, they will be having a plasma membrane. And inside the plasma membrane, there will be a gel-like fluid, which is called as cytoplasm. cytoplasm. So except for this mycoplasma, okay, rest all the prokaryotic cell has what? Cell wall. Every prokaryote has a cell wall. But they do not have a well-defined nucleus. That is why they are called as prokaryote. So they lack a well-defined nucleus. So whatever genetic material is there, that is nothing but the DNA. It will be simply in a circular form, jumbled form. Okay, so they will be open. So hence there will be no nuclear membrane as well over here. So apart from this, what do you say, circular or naked DNA, it has one more thing which is called as what? An extra chromosomal material. Extra chromosomal material, which is a circular DNA in this. Okay, which is called as plasmids over here. Okay, so basically over here, and now depending upon whatever the cell wall or the cell envelope which is there, that is outer covering, whether it is going to stain by some dyes or whether it's not going to be stained. On that basis, there are two groups over here. Okay, that is called as gram positive and gram negative bacteria. So these two groups are based upon whether the cell wall is going to take the stain or it is going to reject the stain, depending upon that. Okay, so if the gram staining is positive, that is violet color, if you see the microorganisms, if they are in violet color, then we say it's what? Gram positive. But if they are in pink color, we say it's gram negative. Okay, so depending upon the cell envelope taking which type of stain, on that basis, two types we have, that is gram negative bacteria and gram positive bacteria. Okay, so cell envelope or the cell wall is very important organelle or a part in the prokaryote cell. So now what are the things which are present in a cell envelope? Okay, so the cell envelope basically consists of three layers. Consists of three layers. So the first layer is called as glycocalyx. So glycocalyx means like in different bacteria, different types it will be present. Like for example, sometimes it may be present in a capsule form, okay, which will be very hard to break. Or sometimes it may also be present in what? Slimy layer. Okay, so this glycocalyx will differ according to the bacterial type. Second type of layer which it has is the cell wall. So mostly the cell composition is of the peptidoglycan. Okay, so if the peptidoglycan is more, we say it's a gram positive. Whereas if the peptidoglycan is less and if it is having lipids, that is fats in it, then we say it is a gram negative bacteria. But cell wall mostly it will be made up of what? Peptidoglycan. So this in turn gives what? Structural support to the cell. Structural support to cell. And the third one is the plasma membrane. So this is the, like if you see this plasma membrane will be selectively permeable, only few things it will allow to pass through it. Permeable in nature. So these are what the three important layers of a cell envelope. Then coming towards the second thing, which is the mesosome. So mesosome in the diagram itself here, I showed you this thing. Okay. So the small vesicles or extensions you can say inside the cell from the plasma membrane is nothing but the mesosome. So what is a mesosome? They are nothing but the membranous structure which are formed by the extensions of plasma membrane as, now there are three types, okay, like how it may be formed. Either they may be in vesicle form, tubules, and lamellae. Okay, so the basic the function of the mesosome is what? Cell wall formation. Then it also increases the surface area. Increases the surface area. Okay, also helps in basic activities for the cell like respiration and secretion. Respiration and secretion. Okay, then the next component is the chromatophores. Chromatophores. Now, what is chromatophore? Now, initially we saw the examples like mycoplasma, bacteria, or the BGA, that is blue-green algae. So, blue-green algae means itself, it will be having what, some kind of pigments in them. So, prokaryotic bacteria means some of the bacteria, they will have what? The pigment-containing membranes. So, those are only called as what? Chromatophores. So, chromatophores are what? Pigment containing membrane. Okay, an example you can take is cyanobacteria or the blue-green algae. Then the next structure of the prokaryotic cell. So I told you three important structures, that is flagella, villi, and fimbriae. So the first one is the flagella. Okay, so flagella is nothing but the thin filamentous extension which arises from the cell wall. Okay, so usually the flagella has some protein which is called as what? Flagellin, which gives a support or strength, okay, basically for the mobility when it's moving, that is mobility. Okay, so this also has a hook and a basal. Okay, so it has two parts, that is hook and basal part or basal body. Like how the tail is, like from the starting how it is little thick and to the end how it becomes thin. So that is nothing but the hook and the basal body. Second one is the pili. Okay, so pili is basically elongated tubular structures. Okay, and this contain a protein which is called as pillin. So, like if you compare the pili with the flagella, this pili will be non-filamentous. Okay, so flagella usually gram positive, gram negative, both bacteria will be having. But pili, it's exclusively for the gram negative ones. Okay, so pili is basic, like apart from moving or like rather than moving, you can say it helps in adhering, means attachment to a host cell to take the nutrition. For that also, pili is being used. Also, it helps in what? Conjugation. Okay, means transfer of the DNA from one bacteria to the another bacteria. For that also, the pili is being used. Okay, then the next one, which is the fimbriae. The small hair-like structures. Okay, they are usually, they will be like the brush bristles, the toothbrush bristles. Okay, so they are basically the bristle-like. fibers. Okay, this also basically helps in attachment. Okay, so pili and fimbriae, both are for the attachment. Flagella is mainly for the movement, but pili also helps in the conjugation. That is the transfer of DNA from one cell to the another cell. Okay, so in motility, like basically pili and fimbriae, they don't have any role in the motility. So if they ask which structure is for the motility, it is only one thing, that is the flagella. Okay, then the next one, next structure or component, that is ribosomes. So in a prokaryote cell, the ribosomes are 70S. Okay, that is 50S plus 30S. So the basic function of the ribosome is protein synthesis. So it synthesizes protein. So either it may be present as a single one or it may be in group. So if it is present in group, then we say it is polyribosome or polysome. Okay, means when several ribosomes come together, we say it's what? A polyribosome or polysome. So basically that is a single mRNA which comes together. M stands for messenger. And RNA is nothing but the ribonucleic acid. Okay. DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. Then you have certain small structures present in them, which are called as what? Inclusion bodies. Okay, so inclusion bodies means nothing but they will be having certain vacuoles such as gas vacuoles or food vacuoles plus glycogen vacuoles. Okay, like that many vacuoles will be present for the storage of certain things. Gas vacuoles for the gas, food vacuoles for the food. Glycogen, the granules will be for the, what do you say, storage of the sugar, glycogen. Okay, so these things are again found in the cytoplasm. Okay, all these organelles you will find in cytoplasm only, ribosomes and inclusion bodies. Okay, plus they do not have any sort of membranes on them. No, not any membrane present on them. Okay, so these are what certain components which are present in prokaryotic cell.